My name's Carrie Damon. I'm the Director of Entrepreneurship Education here at Mars. We are totally delighted to welcome our speaker tonight, Dr. Stuart Foster, who is the founder of VisualSonics, Inc. He's currently a senior scientist at Sunnybrook Health Sciences Centre and professor in Canada Research Chair in Ultrasound Imaging in the Department of Medical Biophysics at the University of Toronto. His current research centers on the development of high-frequency clinical and preclinical imaging systems, array technology, intravascular imaging, and molecular imaging. He is also a fellow of the American Institute of Ultrasound and Medicine. So about the VisualSonics, he's the founder and former chairman of the board and CSO of VisualSonics, a company that was aimed at preclinical imaging, which I'm sure you'll hear about a lot today. He also co-founded the Mouse Imaging Center, now at the Toronto Centre for Phenogenomics. And he's previously been the recipient of many awards, including the Edie Medal for major contributions to engineering in Canada, and the Manning Award of Distinction for Canadian Innovation, and the Ontario Premier's Discovery Award. He also serves on numerous boards, advisory bodies, and is currently on the editorial boards of Ultrasound Imaging and Ultrasound in Medicine and Biology. Please help me welcome Dr. Stuart Foster to the stage.
Thank you very much for the very kind introduction, and it's a pleasure to be down here at Mars. I'm frequently downtown because I have many collaborations here at Princess Margaret Hospital and at other institutions. And it's been my... Yeah, thank you. So it's been a pleasure to work here in the academic community in Toronto, but also to take a small detour, which began about 15 years ago, to get involved in entrepreneurship. And so that's what I'm going to talk to you about today. The company I started is called VisualSonics. It's a biotech company that sells imaging equipment, and we're going to get into the gory details shortly. I know that when I started off as an academic, I had a nice, safe job in a comfortable lab and was reasonably well funded. There was no real need to step outside. But I had to overcome that fear initially because there were things I wanted to do that I couldn't do with the existing technologies. And I thought I had an angle on how to improve that. So really, that's how I got things started. I want to take you back to the beginning of VisualSonics. The company was actually founded in 1999. This is well before Mars Innovation was conceived of or built. There was no Entrepreneurship 101 to help me get through the hurdles of trying to build a company from scratch and trying to attract investment, trying to make sales and make the company successful. So it's been a bit of an interesting journey. And I would say that in my case, a subtitle for this presentation might have been How to Do It the Long Way, because I did take the long road to get to the end. And that's in contrast to many of perhaps to some of the other stories that you've heard at this session. I know I read about a young man whose name is Kevin Systrom recently, who founded a company recently. Does anybody know who he is? There must be a flicker of recognition somewhere. He started a company called Instagram, which, as you know, recently sold for $1 billion. He founded the company something like 5 years ago, but most people think he did everything in the last 18 months. In fact, he got his first funding in the last 18 months, a very small round of $50,000, and things mushroomed from there. I mean, that is a very interesting story. Mine, I think you'll find interesting because it's a different perspective. I'm a research scientist. I spend my time in the laboratory, and I'm not a business person, but I think I managed to pull my weight as things went along. So today I'm going to go through a brief description of the technology that we developed. I developed together with my graduate students and all the people who work with me in the lab up at Sunnybrook. I'll just go through a few selected applications because I think they're interesting, and I still enjoy looking at them, to be quite honest. I'll talk to you, then I'll go back and talk about what I perceived of as being the opportunity way back then. What was it that kind of tweaked me to want to jump into this and try and do it. And then talk about the road that has taken me from 1999 to 2012, and offer a few thoughts on innovation and the sorts of things that I think are important along the way, and that you might be actually wondering about if you're interested in getting into entrepreneurship. I'm going to talk a little bit about physics. This is the first and only equation for you to digest. It's a very simple equation. It has to do with wave physics. We use waves of various sorts, X-rays, optical waves, ultrasound waves to make images of things. And the thing to remember about wave energy is that the resolution that you're going to see in the image is roughly equivalent to the wavelength or some small multiple of the wavelength that you're using. So in ultrasound, the wavelength is a critical factor. In this equation, c is the speed of sound. Many of you will know that the speed of sound in air is about 340 meters per second. In water, it's about 1,500 meters per second. And you also will know that ultrasound works at very high frequencies in the megahertz range. So if that number c is 1,500 meters per second, or 1.5 millimeters for every millionth of a second, and you have an oscillation at 1,000,000 cycles per second, if you do that division, the wavelength is 1.5 millimeters. So wavelength governs the resolution that we're going to see in an ultrasound image. It is absolutely critical. So as we start looking at those frequencies and looking at the resolution that you can achieve, all of the diagnostic applications are in this range, somewhere between 1 and 10 to 15 megahertz. The wavelength there ranges from, as I pointed out, at 1 megahertz, 1.5 millimeters, to 150 microns at the higher frequencies. So that tells you what the resolution is going to be. When you go down to do an OB exam, it's going to be approximately a fraction of a millimeter resolution, and that's because of this frequency range. So in ultrasound imaging, we talk about axial resolution and lateral resolution. Axial resolution is the resolution along the direction of the beam. Lateral resolution is the resolution across. It doesn't matter to distinguish between these two too much because they're fairly similar. But if one increases the frequency, say to 20 or let's say 50 megahertz, all of a sudden your resolution can be on the order of 30 microns to 60 microns. So that's exactly the resolution I need if I want to image a mouse, for example. And we'll talk about why I might want to image a mouse in a second. But what's critical here is that the technology for doing clinical ultrasound imaging was confined in that 5 to 10 megahertz frequency range because no one could build a transducer, effective transducer, to work higher than that. And that's essentially the problem that we solved to create the technology that I'm going to describe. We figured out how to make very complex array transducers in that higher frequency range so that we could turn up the resolution. I'm not going to say a whole lot about the technology, but the result is that we were able to create transducers, which are shown across the front in this picture, with a range of frequencies from about 20 to 60 megahertz. And by doing so, we created an opportunity for ourselves to do what I call microimaging or micro-ultrasound in very small regions of tissue. So it could be human imaging or it could be imaging of a small animal. And I'll tell you why we chose mouse imaging in a few minutes. But to go along with these transducers, which incidentally each are worth sort of an entry-level BMW in terms of cost, we need another system that's going to actually turn the signals that are coming back from all of the individual elements in that array into an image. That's called a beamformer and then a console for the display. So that's all over on this side of the page. And that's the other requirement for us to make micro images. You need a nice way of using the ultrasound energy that comes from the transducer, and then you need the beamformer to create the picture. So what would I do with such a scanner if I had it in my possession? And this was really the insight that I had back in the 90s, is that actually there might be people who would want to use such a piece of equipment to solve very interesting problems in biomedical research. So I'm going to show you a quick video. It's not too long. And it's going to talk about the development of an anti-venom construct to be used on box jellyfish. So if you go surfing in Hawaii or in Australia, or if you've ever gone scuba diving off Australia in the Barrier Reef, you'll know that box jellyfish are a very dangerous animal, and it would be very nice to have an anti-venom because people end up dying on the beach. So enjoy this short clip.
Jelly. And we have calculated our doses based on the size of the animal for a dose equivalent to a fatal human sting.
It's easy to kill a mouse with box jelly venom, but quite another to bring it back from the brink.
We're building tension.
First, the mouse is anesthetized. Then it's laid out for the test, with its chest shaved.
So here you can see the transducer in the foreground, the mouse
ultrasound
obviously.
machine will monitor its heart rhythms as the quick-acting
There's the
toxin
scanner.
is administered. Followed by what they hope is the life-saving blocker.
This is about as nasty as it gets in the natural kingdom. We can start to unravel the mechanisms that this venom is using in the death of a mammal, a mouse here, or a child in North Australia. Ready? 306:15.
They inject the venom first, and within seconds, it begins to take effect.
You could see it's already affecting it. It's already changing.
The heart speeds up and begins to beat in an erratic way, quivering more than beating, pumping no blood.
You have a hypertensive crisis. The way the heart muscle is super clamping, that's a hypertensive crisis. So this is recapitulating a death on the beach when you have a human that dies in 5 minutes.
Until now, there would be no way for a mouse or a human to recover from this much venom. But now Angel administers the trial blocker.
Okay, go. All right, so we are 66 seconds out.
At first, the blocker seems no match for this amount of venom.
We're pushing it. He's having a hard time, but sort of on the edge. We're hard to call it right now. I mean, there definitely is a positive effect by the blocker, but will it be enough to save this mouse?
But gradually, the heart shows signs of returning to normal function.
We don't look like we're on death's door, so we're good.
Finally, the heart is recovering better than they could have expected.
This is great.
You can see the actual heart.
Oh my God. It looks very good. We see normal heart function at this point. Well, that's fabulous. We've saved a mouse. So he's alive.
But can this same blocker save a person from a venom so toxic and fast-acting? Angel is well on the way to creating a breakthrough remedy. And soon, perhaps, the terror of the box jelly may have lost its sting.
So the terror of the box jellyfish may have lost its sting. It's an interesting segment. That played on Nova on the PBS network in the United States. And it's very similar in terms of the development of applications for the instrument in other areas of investigation. So for example, in the developmental biology field, people want to know how embryonic development happens. What are the important genes that are involved? How do knockouts of genes affect the development of an organism? How can new therapies be developed to change the way development occurs? The field of cardiology, what happens when the heart goes into a myocardial infarction? Vascular disease, the buildup of atherosclerosis, cancer, neurobiology, inflammation, and the list goes on and on and on. So around the world, people are using technologies like the one I just showed. Obviously, ultrasound is only one component in this whole story, but they're using these technologies to follow animals as they try to study these issues. Well, this is interesting. I'm missing oh, there it is. So I'm just going to quickly go through a few pictures. So here is a picture of an embryo. This is a day 13 embryo, a mouse embryo now. So the field of view here is only about 10 millimeters on a side. Obviously, you can see the heart beating here. You can see the chambers of the heart, the head of the mouse, and the torso. Ultrasound, one of the advantages of ultrasound is it's very fast, so you can do real-time imaging with it. You can also, for the mouse mother, provide a nice three-dimensional image of the mouse babies, as long as they pay a certain amount of money. One of the other interesting things that you might want to do would be to intervene in some kind of biological situation. So, for example, if we're given the goal of injecting, say, some DNA or some other active ingredient into the spine of a day 12 embryo, the spine is shown right here, how would you do it? Well, you can use ultrasound to do guidance in that case. So we play here, and you can see the needle going through the different membranes and entering the embryo, and finally, in this very deformable tissue, finding its way into the spinal canal. Then you could introduce some viral vector or DNA or whatever the target was, or you could extract tissue. So that's quite helpful to people. You can look at cardiac function quite effectively. So here you see a beating mouse heart. And one of the great things about ultrasound is that it's blazingly fast. So we can operate at speeds of up to 1,000 frames per second, and with some effort, we can go up to 5,000 or 10,000 frames per second. And as you know, mice have very rapidly beating hearts, so we have to slow that down so you can study the biomechanics effectively. But you can see here the myocardial wall, the thickening and thinning as it goes through the cardiac cycle. You can identify the chambers, the left ventricle, the right ventricle, aortic outflow tract, the apex, and so on. And then you can make quantitative measurements on these things, and you can investigate these when there's been some kind of an intervention. So, for example, here we're looking at two mice. One is a healthy mouse with a healthy heart. You can make a measurement of the ventricle. You would do this in a dynamic way, not just on a still frame like this. And you can do this on an infarcted heart. So here, surgeon has gone in and actually occluded a coronary artery. The heart wall has atrophied, and now this mouse has a myocardial infarction. And this becomes a model, of course, for scientists to use to try and develop improved therapies, to find ways to get that wall to regenerate and so on. You can look at strain, because ultrasound can see these dynamic changes that are going on in the heart. So the heart has a kind of torsional motion, which gives circumferential strain, a longitudinal motion, which is shown in this direction, and of course the radial strain if you look at a short axis view of the heart. Just quickly, to show you the kinds of things you can do, you can tag the borders of the heart muscle, watch all of this happening dynamically. In this case, we're plotting the velocity vector on the myocardial surface, on the inside surface of the heart, and looking on the right side of the screen at a loop, which shows how that little pixel of heart muscle has moved around during the cardiac cycle. And you can imagine that if the heart wall is compromised, these things change. So now people have a quantitative way to look at exactly what's going on in the heart, and that's quite helpful. We can also make agents to inject into mice that actually have very interesting properties, so-called contrast agents. And the agents that we inject are microbubbles. So these would be 1 to 2 micron-sized bubbles that are injected into the bloodstream. They essentially are like surrogates for red blood cells. And the great thing about microbubbles is they ring like a bell when you hit them with ultrasound, and they send back a very special signal, a non-linear signal that we can detect. So interesting things we can do with these bubbles is that we can actually put molecules on their surface. We could put a ligand, for example, for some target, like the VEGFR2 receptor, and send these bubbles into the bloodstream, find out where VEGFR2 is being expressed. And we're actually doing studies like that. I don't have time to go into the details on those things. Here's an injection of microbubbles into a tumor. This is a tumor model. This is growing on the flank of a mouse. You can see the dimensions, but on this side, you see a cloud of microbubbles entering into the tumor. So this just tells us how this tumor is filling with blood. This is the raw image, so we can become a little bit more sophisticated, do an overlay, and watch that wash in. Now you see a projection of all that washing in of blood into this particular tumor. Now, if you were going to give this mouse an anti-angiogenic therapy or a chemotherapeutic agent or a combination of the two or any other cancer therapy, you could ask the question, how does the therapy affect the flow of blood? Are we choking off the blood supply like we would like to, or are we promoting it? In fact, there might be good reasons to try and promote it, believe it or not, but I don't have time to go into that. And then we can actually quantify those things to look at perfusion into the tumor. So here's a real-time view of a tumor filling with blood with a quantitative measure of perfusion. And so you can see in that nice colorful picture the regions that are well perfused in the shades of orange and yellow and poorly perfused in blue. But you'll notice that this tumor fills eventually. So all parts of the tumor are actually getting blood. Another example in the brain, and this has been a very interesting area of development recently, is using that contrast, inject it into the brain, and look at how it spreads through the cortex. And this is the work that Lise Lindfear, who's sitting right down here, was involved in with a postdoctoral fellow in my lab. And the postdoc's name is Martin van Rijs, and he has just recently returned to the Netherlands. But you can see in this picture how we can follow the flow of blood in a very detailed way in the cortex of the brain. And then we can do reconstructions of these vessels. And here's an example of a reconstruction of the surface vessels in the rat brain that have been painstakingly segmented, and of course with fancy 3D graphics now you can look at all those perforating vessels in the brain in a very interesting and quantitative way. We can also do functional measurements in the brain. I don't have time to actually show that to you because the available time has been used up for showing you pretty pictures. And I want to talk about how I arrived at this place. And I just want to point out that even back in the late 1990s, you have to remember that this was a time that when there were no targeted drugs for therapies of cancer and this sort of thing. There may have been one or two drugs that had been released at the end of the 90s, but it was just sort of beginning. And so this idea that one could develop targeted therapies was just emerging. The other thing that was happening at the time was the fact that the human genome was being sequenced. And the research community knew that they were going to finally have the code and all of the genes sequenced, but they knew nothing about what these genes did, or very little about what they did. So they've also begun the process of systematically knocking those genes out and asking the question, what is the phenotype of my mouse? And of course, what do you need to look at that phenotype? Well, if you do a knockout experiment, the last thing you want to do is kill the mouse that you're trying to examine. So you might as well start using some advanced imaging technologies to look at those mice. And so that's exactly what happened. But at any rate, just addressing this slide, of course, this is a long process. It takes a long time to go from a biological discovery at one end of this slide through target identification, through testing in animals, 3 phases or 4 phases of clinical trials, and then finally down the pipe into what we now call personalized medicine. It is a very long process, but being able to work more effectively in this area, in the preclinical stage, we felt was going to be very important. In fact, it was a bit of an epiphany in the late 1990s to wake up and say, yes, imaging is important for people and for these clinical trials, but it's equally important back here when you're trying to look at the animals that we use to help us drive the directions that we're going to go in for our clinical studies. So I and a few other people around the world began developing these technologies. Of course, I was working on the ultrasound technology. Other people were working on magnetic resonance imaging, PET imaging, SPECT imaging, optical imaging, you name it. It just sort of happened at that time that we knew we had to do this. So all of these imaging technologies were developed, and they all have a place to play, or they have a role to play in the current preclinical imaging field. The particular advantages of ultrasound over some of the other techniques are: 1, it's very fast; 2, it's reasonably inexpensive; 3, it's portable, easy to use in a laboratory setting. You could bring it into a animal colony and work with it there. So that's why we worked on it, and that's why I got started on it. And I came in one day to work, 1997 or early 1998, and said, we're starting a new project today. We're building an ultrasound scanner that's only going to do one thing. It's going to image mice. Only later did I start to think that maybe I need to start thinking about how this might turn into a business. And the reason I did that is because colleagues were coming to me and saying, well, can you build me one? Because we want to look at XYZ gene. Can you build me one? Because I need to study the heart. And at the newly founded Mouse Imaging Center, we wanted a couple down there so that we could do our cardiovascular investigations. So people wanted scanners. I was a laboratory. I was not commercial business to build scanners. And so I thought, this would be a great place to start to develop high-frequency ultrasound, not just for the small animal market, but for the clinical market as well. But I'm going to start with small animals, because I can prove my technology in that sphere, and then once it's become successful there, I can transform it into a clinical product. So what about that market? Well, there are $27 billion a year spent on instruments to do bioresearch. That's a lot of money that's being spent by NIH, other agencies, drug companies. Some of the companies that produce that equipment are listed here. These are the big companies that you people would be familiar with. They're very successful. And what they do is they provide equipment for the drug companies and the manufacturers and the people developing new therapies to actually evaluate, test, and to develop their products. Well, that's a lot of money, but how much might be available for me in small animal imaging? Because that's where I want to start. And so we worked out that there was somewhere on the order of, on the order of a market that we felt that would be about $300 million. That's since been verified. These are 2005 numbers, and it's grown since then. So that's the whole market for magnetic resonance ultrasound, everybody to play in. But it's growing pretty rapidly, roughly about 10% per year, which is reasonably rapid. In the early days, it was growing at 40% per year. And in 2005, ultrasound share of the market, this is 3 years after we introduced our scanner, was 9%. So it was growing and it was developing. The great thing about ultrasound is we didn't have any competitors. And that's not true of the other manufacturers. MR, PET imaging, micro CT, there are all dozens of companies working in those areas. We also know there are 6,000 animal labs scattered around the world, and they're in universities, hospitals, research institutes, pharmaceuticals, biotechs, contract research organizations. They have need for this kind of equipment as well. So we knew that we had a reasonable number of institutions to target. And not only that, within each individual institution, there are multiple opportunities. I know, for example, in my lab, I have 5 scanners right now, and I pretty much need them. And now I have to share them with other people. I don't like to do that. So you may need more than one in each place. Sorry. So the other thing that is important is that the instrument has to develop over time. You have to start with something that's very crude, and then it has to become sophisticated. Of course, in 2002, when we developed this scanner, we thought it was the best that there would ever be. But it only provided 4 frames per second. That wasn't too good. We knew we had to advance, and clearly from a commercial point of view, you have to be doing leapfrogging. So it wasn't long before we had new technology, 2004, a vastly improved scanner that could actually do 100 frames per second. 2008, the cream of the crop of scanners, at least by today's standards, which used that array technology that I talked about earlier. So this is the current generation of scanning equipment, which has now been combined into a super scanner that can do photoacoustics, which is a combination of optical image and ultrasound imaging for preclinical applications. So that's a good evolution over time. And in fact, you can expect the next generation in a couple of years, and it is going to be spectacular. But I can't tell you what it is. As you're developing a company, of course, you need revenue growth. That's critical. If you don't have it, you're going to fail. I was astounded, when we first started selling equipment that we actually had any revenue. That was exciting to get revenue. And in fact, most of the startups that were my competitors or who had been co-funded at the same time had no revenue at all. And they're sort of working on vapor, but there's an art in doing that as well. But at least you can see over the period of time from 2000 to 2012, there's been pretty steady growth. And so now we have revenues of about $35 million a year, and it's continuing in that growth cycle. You can also see that when we introduced new scanners, things happen. 2004, that was that new scanner that could do 100 frames per second. Whoa, it shot up. A little bit later, when we introduced the scan, you see that's starting to fade in 2008. Then we introduced the next scanner, and it's starting to shoot up again. And of course, we hope that this will continue. Of course, you never know, but we're optimistic about that. And the other thing is that EBITDA, which is a term that essentially describes the income of the company before you take all the taxes and stuff off, became positive in about 2009. And when you're talking to investors, that's a number they really care about. They really want to find out about what your EBITDA is. And if it's positive, that's a very good indication that you're a healthy company. So we were a healthy company. However, if I could just go back to this, we're talking about 10 years. I guess Jeff Courtney isn't here anywhere, is he? Jeff was the VC who put a lot of money into VisualSonics. The early days, Jeff would tell you, that's not good enough. You got to grow faster than that to make my day. But that's what venture capitalists are supposed to do. At any rate, 10 years. So this cartoon reads, and you, Al, what would you do if you had to wait 10 years for a liquidity event? That's a real problem. Okay, VisualSonics' fate. So we did have a liquidity event. Now for me, a liquidity event is when I go to the bar and have a few too many beers. But for venture capitalists, it's selling the company. So VisualSonics was sold in 2010, and it was sold to a company in the United States called Sonosite. They're a very large manufacturer of ultrasound equipment. They're a $1 billion company. VisualSonics is closer to the $100 million area. And they absorbed VisualSonics because they saw the future of this technology was in clinical ultrasound, and that's where they're taking it. And they were willing to pay a premium for what they consider to be an important asset for their company. Sonosite is just another fish, really. They were purchased just a few months ago by another company called Fujifilm, who most people don't think of as being a company in the healthcare business, but they are, and they're a multi-billion-dollar company, a Japanese company, obviously. But they have designs on all of this technology across the board, from preclinical to clinical. So they were looking out there, and it's one fish after the other. And but that doesn't mean that we shut down the office of VisualSonics here in Toronto and close it up and send all the jobs to Japan. Because the one thing that we have here in Toronto that they don't have there is the know-how. So they need that. And VisualSonics is quite happy and healthy up at Lawrence and Young producing ultrasound machines. So let me spend the last little bit of time, I was told to leave at least 10 minutes for questions, going through some of the issues that I saw along the way and what I think is important. A lot of these things from an innovation point of view just look self-evident, and it's probably true, so I'll go through them quickly. You do need a good idea to get started, but you need to be invested in that idea, and you need to be able to champion that idea to no end. You have to be willing to go to the end of the earth to make it happen. So if that means going to 5,000 VC boardrooms, that's what you're going to have to do. You do need some intellectual property, but I would suggest probably not as much as you think. You do need to incubate it in the lab because you need to make something work before you can get it out there and get the respect that you should have to get funding for it. You do need to have identified a real market, and by a real market I mean problems that your instrument or your device or your therapy is or are going to solve. And you also need good contacts. You need to talk to people. You need to network. You need to be at meetings like this to talk to people who've been through this or others who are interested and have other angles on what you should be doing. Getting going is very simple. Start your company, incorporate. You become the president, you become the secretary, the CEO, the everything in the company. And then you set up a share structure, you appoint a board of directors. Don't appoint your mother to the board of directors. It's not going to be helpful. You have to determine who the equity holders are going to be. This is actually quite important, and thinking back, I think I did a reasonable job, but think long and hard about this and then start hunting for money. So what do you do with all that equity? Well, you're going to have to share it. So there's the founders group. I wish this thing would stay on a bit more. There's the investor group. There are the host institution. They all want a piece of the pie. They all want in. You have to remember, it all adds up to 100%. There's no more at the end of the day. And you need to keep some in reserve because you will need to attract some special talent to your company. Don't give it all away. I say the VC, you have to respect the VCs. These people have what they call skin in the game. They're putting up real dollars, they're putting it on the table, they're paying for things. They deserve equity. Host institutions, on the other hand, I think should not be in this business. I don't think they should use equity from their startups. They should rely on royalties, licensing agreements, this, that, and the other thing, but not equity, because that equity is needed later on. Business model, you'll have to develop your own business model. It all depends on exactly what you're trying to do. Of course, you have to have a good product. It's essential. In my case, I think I had a pretty good angle. My angle was, I'm going to develop a preclinical product, but I'm going to build it to FDA standards. So when the time comes for it to become a clinical product, I just have to step across the street and go through that regulatory process. By doing the preclinical side, where there's much less regulation, I was able to skip through a lot of those really tricky things that go on when you're trying to develop a proper medical device. And now that regulatory hurdle will be passed, but it won't be passed by me, it'll be passed by those larger corporations. You have to provide the end user with a complete solution. Part of the genius, I think, of the business plan of VisualSonics was not just dropping a scanner box off at the loading dock and saying, go to it, guys. VisualSonics did everything. They provided the rail system that the mouse would sit on. They provided the instrumentation for monitoring the mouse. They provided the anesthesia system. They provided people to go out and help you learn how to use your scanner. They provided tumor models. They did everything from start to finish. So that when a person ordered a VisualSonics product, they knew they would actually be able to get the job done that they actually started out to do. Quality assurance is absolutely paramount. If you don't believe that, you'll be out of business in a month. Because when you deliver something, it's got to work. It can't die on the loading dock. If it does, your whole company is going to die. You have to maintain a strong customer focus, which means when things go wrong, you're out there to fix it. No ifs, ands, and buts within a few minutes. Don't lie to anyone. You can't exaggerate the capabilities of your imaging system. You can't lie to investors. You can't lie to your colleagues. It's just bad practice, and that also will put you out of business. Sorry. Be careful about the numbers you write down in operating plans. I remember sitting in the boardroom for VisualSonics, and the goal was 60 scanners, and they had 52. I thought, great, not too bad. We're close. The board members went crazy. That's the kind of rigor they demand. If you say you're going to deliver 60 scanners, you better deliver 60 scanners and no less. If you're going to put a stake in the ground and say you're going to accomplish something by a certain date, you better have it done or your investors will lose confidence. Be very careful about that. Develop a company culture. Well, it's very important to do that. And so our company culture was defined by our president, whose name was Tom Little. I'll introduce you to him in a few seconds. I'm not going to read this whole list, but innovate without ego. Don't let egos get into the picture. Find out how you can, not why we can't. Where's my pointer? Be intolerant of bureaucracy that adds no value to the customer. It's just not helpful. Team values. So this is really to define how you try to interact in the company. I'll just read a couple of these again. Honest communication, no held punches. Debate ideas openly, debate passionately, then get on with things, make decisions. 100% buy-in, that means no unresolved issues, no politics. You leave the room, everybody with the same decision. Finally, of course, it's not a democracy, as Tom will point out to you. The company is going to make decisions, and you go with them. That's the way it goes. It's not a democracy. IP, well, the more the merrier. It's nice. It's a security blanket for the investors, pretty much. It's an important part of valuation. It's one of the things they will definitely look at. You want to do it. It's a bit of a pain, and I'll talk a bit more about that in a second. VisualSonics, when I started the company, I had one patent that was irrelevant. Okay, one irrelevant patent. But the thing I did have was know-how. And I was able to demonstrate that know-how to the investors. And it's interesting to note that we had no competition back in 1999, and we still have no competition in 2012. And that's because we guard that know-how. And that's a very important point. At any rate, it is important to develop the IP portfolio. VisualSonics has not slacked off. 38 US patents and 100 foreign patents at the present time. People are the most important part of the company. That won't come as any surprise to anybody. You have to retain key employees. And people are finicky. You got to work with them. And people think they're valuable, and they are valuable. And you have to use some of that equity that I talked about before to keep them in the company. And you have to be the judge of how much is needed to do that. It's a difficult struggle to find out exactly what that is. But I mean, you keep the key people at all costs. So whatever it takes, you have to do it. You have to keep real business people around to manage the company and sit on the board. This is not a game for amateurs. It's not a game for scientists. It's not a game for researchers. It's a game for business people. And if you don't do that, you won't survive. 1 or 2 scientists on the board is enough. I think I'm going to bring this down to 1 scientist is enough. You don't need scientists on the board of directors. The board of directors is to run the company, the corporation. It's not to decide on good science or what sorts of things we should be doing in a scientific way. It's to make money. Their goal is to make the organism, the corporation, successful, self-sufficient, and bringing lots of money in. Hire the right CEO. This is the most brilliant decision I made in the whole process. I hired a guy whose name is Tom Little, shown here. Yes, this picture is a little bit of a joke. World's best boss. But Tom was a driven man. He never let up for 1 second. And he defined the company culture, and he brought in key people who are going to make things happen within the corporation. This is the most important hire you'll make. If you hire someone who's halfway there, you're dead. And you'll notice I've been saying you're dead all the way along through this conversation. If you make any of these mistakes, you'll be dead. Board of directors. Hire real people who are smart. So here we have Sam Maslach over here. He founded a $1 billion ultrasound company called Accuson that was sold to Siemens. Steve Nakashige, the former president of Hologic, which is a large digital mammography / X-ray company. Some, I guess, scientist person. Sam Iffergan. Sam was the first guy who came into the lab and said, wow, can I put money into this? And he did. He put $1.5 million in at the very beginning. This is what got us going. Our venture capital friends, Jeff Courtney and a person who's in Graham, can't quite drag out his last name. They were both with Vengrowth. I got funding from Vengrowth. And Tom, who's in the background. That was our board of directors, and that's what built the company. Yes, we needed money. So we raised some money. We raised $13.5 million to get VisualSonics going. I'm going to go more quickly. Control is important too. So I founded the company. It's my company. I want the whole company. I don't want anyone else involved. I got to control everything. That's not the right way to do it. You have to let it go. You keep as much control as you can early on, and then you loosen your grip but you try to keep your influence intact. And you need to avoid this thing called founder's disease, in which case the founder is trying to dominate everything and it just doesn't work. Graft versus host. This is an interesting disease. This is a disease that develops between an academic community and a commercial community. They don't mix. Don't try to do it. It doesn't work. Initially, I thought I might be able to pull it off, but I can tell you from experience, I failed. I failed at that. It doesn't work. So our view really for commercialization, this is not just for VisualSonics. This is for 3 very exciting startups that have been very successful at Sunnybrook. So keep your commercial people at one venue. This is on Yonge Street. This is where all the commercial people are. VisualSonics, Sentinel, Profound. About 250 people. Ultrasound and MR. Just commercialization. No academic role at all. Get them to send their money over here to Sunnybrook, where we have our labs and we want to do research. And you can work out the details of that. About 250 people, 75 trainees. Many of the imaging tools, basic and applied research, just an academic site. We just train students and try to do the best possible research we can. Reasons for success? I think we were successful because the ideas we came up with were not invented overnight. We worked for decades on these things. I started working on high-frequency ultrasound in the 1980s. The founders were international leaders in their particular areas of research. We were driven by the problems and in fact became our own consumers. We knew we needed to solve a problem, we knew what had to be done, and we were willing to buy the instruments. Recruit excellent people, I already said that. Patents are important. Know-how is essential. Provincial matching programs are very important, and SHRED tax claims or tax rebates or whatever they are very important as well. Without them, it's touch and go, to be quite honest, because you'll be using all of your money for more important things. Focus on business and markets, not science. By the time it gets into a company, it's business, not science. So here's my report card that I've given myself. I give myself an A+ for the prerequisites. I thought we had good ideas, and I thought we did a good job of putting at least some kind of prototype out there that people could see the value. Equity, I thought I did reasonably well at that. I held off Sunnybrook to 5% of the company. I think it should have been 0. The business model, I think, was excellent. We really focused on making a solution for customers. IP, B, okay, I admit I wasn't too hot on patents. People, A+, because the people who are in VisualSonics are real leaders. Tom Little, who was our president, is now the president of Bell Wholesale. That's a 1,000-person organization. Anil Amlani, who is our CFO, left VisualSonics, is now CFO of the billion-dollar corporation that took VisualSonics over. Less emphasis on bureaucracy, less IP warehousing, quit building patent portfolios and expecting to license them. Just get on with getting the ideas working and improve incubator facilities. So I have to thank the VisualSonics gang for initiating all these ideas and doing a lot of all the hard work. My students, postdocs, staff, people who helped me at Sunnybrook. And of course I have to thank VisualSonics as well. The leaders at the company were superb. I've already talked about Tom Little and Anil Amlani. Others like Des Herson, who was the head of engineering, were absolutely essential. Couldn't have done it without them. So where are we going to go with this tonight, Boris? The same thing we do every night, Pinky. Try to take over the world. There you go. Thank you very much.
Thanks, Dr. Foster. I think that your many years of superb research and hard work have really paid off in an incredible success. There I am, yes. Would anyone like to ask any questions? If you could just go to the mic, please, that would be great.
I have 2 questions, one about the technology itself and the other about your development process. Does it only work in squishy stuff, or can you handle solids? I'm thinking industrial non-destructive testing. And the question about the development, I recently read an article on a number of successful entrepreneurial ventures which started out doing something completely different than the thing that they finally succeeded at. And it was simply that they recognized that something they thought was irrelevant was what got the customers all warm and giggly. And so they said, okay, we'll do that, and ran with it. Did you have any similar experiences?
Okay, squishy stuff. In principle, yes, it should work on non-squishy stuff. The principles are very similar. It's a little bit harder in solids. The reason it's harder is there are what are called mode conversions from the propagating wave into shear waves and other more complex waves. But the people in the oil and gas industry use sound just the way we use it, and they are able to map out oil beds and so on quite well. Not perfectly, but they get a very good idea of where the oil is and how it all works. So yes, non-destructive evaluation in principle, yes. It tends to be a smaller market, but maybe we just haven't explored it enough. On the other question, it's a very interesting question. Yes, failure is a part of success. So I had a reputation for messing companies up before I decided to start VisualSonics myself. So when I started my career, it was kind of a badge of honor to be associated with GE or Hewlett-Packard or any of the other big multinationals. I worked with Hewlett-Packard for many years, developed my ideas, and then saw them kind of fizzle out into a bureaucratic haze on the horizon. It was very difficult to move inside big companies. So by 1999, I had finished off a few projects at VisualSonics at Hewlett-Packard that fizzled into the ground, a few other smaller companies that fizzled into the ground, and I decided just can't stand it, I have to do it myself, and VisualSonics is what came out of that. So yeah, you will probably fail before you succeed.
Hello. I recently read an article. It's about U.S. medical device market. And the complaint was that large players, they squeeze out new startups and little guys by using questionable tactics like bundling and kickbacks. Did you face this problem in any markets? And if you did, how did you handle that?
Well, that's an interesting observation. I have to say, having gone through this process from start to finish here in Toronto, I was super impressed with every step of the road. You know, the integrity that the different groups brought into the picture. The venture capitalists, I mean, before, you know, venture capitalists were just ruthless whatevers and didn't want to have anything to do with them. After I was finished going through the process, I had a new respect for how the whole thing works. They have a vested interest in making sure the company is successful too. And you notice I put on my little list of things not to do. Not to lie is one of the things. It'll just get you into endless trouble. But I found everybody to be above board, honest. I had no problems at all going through the whole thing. So I don't know how those situations arose, but it didn't happen to me.
So you didn't have problems with, I don't know, companies selling like similar medical devices and trying to squeeze you out?
No, not at all. And as I pointed out, the reason being is that critical factor know-how. Nobody else could produce these transducers. That was the amazing thing. You know, that we developed a technology that was very difficult to reproduce. And it's true, we tried to build as many patents around it as we could. But really, it's just the physical job of building those things that was so hard. So will we have competitors? I'm sure we will. But I think we've got a good, strong lead at the present time, and we intend to be leapfrogging ourselves as much as the next guy will be. So hopefully that won't pose to be a problem.
Thank you.
As far as the business, especially funding, I noticed in your example, most of your funding, it appeared, came from venture capital. Is it difficult, or impossible, or just not practical to bootstrap such an operation versus going mostly VC?
That's another good question, and I would say yes, VisualSonics did it with venture capital. It's one way to do it. The other way is to develop the company based on investments from another group of people that you're quite familiar with. So let me give you an example. A company that my colleague Don Pluis and Cameron Piran started, it's called Sentinel. They make breast magnetic resonance equipment. Very successful company. How did they do it? They knew a lot of radiologists who had a lot of money sitting around doing nothing, making 2% in the market or something. And they knew the idea was good. Why? Because they're radiologists. Just like we knew this scanner needed to be built because we wanted to use it ourselves. So there are ways to do it, to find communities where they have money and they can believe in your ideas. And I frankly think the Sentinel approach was a good approach, perhaps in some ways better.
Great.
Yeah.
Thank you.
So my question is regarding the CEO that you hired. When did you find out that you need an external CEO? How do you find one who was willing? Did you pay him cash, or was he willing to work on equity, and how do you find one?
Finding CEO is tricky, but here again, the process works in your favor because, for example, I had borrowed a ton of money from the venture capitalists. They did not want to see their money going to pay someone who was not going to get the job done. So when it came to recruiting the CEO, they had lots of contacts. They knew lots of people, and they hired top flight recruiting companies to find people that we brought in for interviews. And of course, eventually we hired Tom because he was clearly the guy who was going to get the job done. So really, they are a big help in that sort of process, and they'll make sure that you hire a good CEO if you're not going to pay attention. Yeah.
How hard was it to go through the technology process of getting it into the market and developing, manufacturing the actual ultrasound?
That's also an interesting evolution. So I can tell you for sure that what we considered to be in our lab, a piece of equipment that was ready to go to work in another laboratory, they just looked at it and said, no. That's not a manufactured piece of equipment. Now we're going to have to put it through this whole quality control cycle. We're going to have to develop a manufacturing process. And to do those things, you have to hire experts in both of those areas. So, and these are high-level hires. That you pay big money to get people who can do this properly. But you have to hire those experts to turn what you consider to be a manufacturing process into a real manufacturing process. So it all has to be done that way. And it has to be done in a very professional fashion. Finding those people is very important as well. So there's a lot that goes on from the laboratory system to a manufactured system. And it's very critical to make that all happen. I find it amazing now to go over to VisualSonics. I know how my lab works. I go over there and I see these assembly lines of things lined up methodically, everything in its right place, things coming together. It's an amazing machine.
Thank you very much for sharing your experience and your expertise and all your insights on how to really build a high-tech company. We appreciate it very much. Please join me in thanking Dr. Stuart Foster.